Hey, as promised, I'll try to explain the mRNA vaccination in a simple and maybe oversimplistic way, but I hope that I will be able to make the main message come across. So, just a quick recapitulation. What is mRNA? mRNA stands for messenger RNA, which essentially means that this messenger RNA is kind of equivalent to the DHL cars that we have here outside, meaning they deliver important information to different places in the cell. So, in some way, you can call them the DHL cars of our cells. But what exactly do mRNAs do? Again, in very simple terms. For this, it may be actually interesting to take a closer look at our general cell structure, or at a very simple example of a cell. Consider this piece of paper here a cell, a human cell, for example, or a cell of any other, like multicellular organism. You have essentially two areas in that cell. One is the nucleus and the inside of the nucleus, which you see this circle, right? And the other one is the non-nuclear area of the cell. Of course, there's many other things in that area out here, but for now it is sufficient to just focus on this very general subdivision of the cell. And what do you have inside the nucleus? Most importantly, there is the DNA. And as you might know, the DNA can be viewed like a ladder that is twisted. And so it's, which is also called a double helix structure. So this twisted ladder has particular steps, and these steps consist of bases that are on each side of the strand of the DNA, and they're always complementary, meaning when you have one type of base on one side, you will always have another specific type of base on the other side. Those bases are essentially molecules, and there's only 4 different types in the DNA. Here, no, 4. So if you already know, or just know one half of a DNA, like one strand with just half of the steps in each case, you can automatically infer the other half, which is actually great for many molecular biology approaches, but also for how cells work in general. Also, what in multicellular organisms happens is that the DNA is always protected inside the nucleus, so it never leaves the nucleus and goes into other areas of the cell. So in order to get the information from the DNA out into the body, essentially, you need to read that information and then bring it outside. And that's what the mRNA is for. The mRNA essentially are small little pieces of a one-strand part of the DNA. Simply speaking, they replace one base with another base, but in principle that's what they do. And through special enzymes, the DNA is opened in certain areas of interest, and then the mRNA copies that part or reads the complementary section of that part, and then is being brought outside. I mean, there are several other steps in between, but it's then being brought outside, and there it meets the ribosomes, and within these ribosomes, those base sequences, which code for certain amino acids, are then translated into amino acids and into polypeptides in the end, which means proteins are being formed eventually. After this process is over and the mRNA is no longer needed, it is being degraded, and its individual pieces, or the different bases and so on, will swim around in the cytoplasm essentially and will be used again for the next mRNA to be built. So these are these general steps can be subdivided into 2 major steps. One is the so-called transcription, which is happening inside the nucleus. Like here, the DNA information is transcribed to an mRNA structure. And the other one is the translation, which happens outside of the nucleus. and where the mRNA is used to generate proteins, to translate that information into amino acids and eventually into protein structures. This whole process is called the protein biosynthesis. So now you know what the mRNA stands for and what the mRNA normally does. And that's important to know because there's one part that happens inside the nucleus, which is like the dangerous, tricky part of the whole thing, because this is where the DNA is, the most important information that we have. And the other part is outside, which is essentially for generating proteins, but which doesn't affect the DNA itself. So what does this mean now for our mRNA vaccines? If we take a coronavirus, like here, you see the following structure. The coronavirus has some special viral DNA inside, and then it has these so-called spike proteins, which actually also look like spikes. And these spike proteins are being used to enter a cell. And eventually, that's what all viruses do, take over that cell, so the cell would produce only more viruses. And what now the mRNA vaccine does, and that's actually a very smart thing to do, is it only copies or takes this genetic sequence of that spike protein, nothing else, and makes an artificial mRNA. I mean, humans do this. One can generate an artificial mRNA. Actually, it's not that complicated. That sequence you can generate on the computer, and then you can basically take all the ingredients that are necessary for an mRNA, and then you basically put it together in a lab. So what you end up with is a specific spike protein mRNA, meaning an mRNA that codes for this spike protein, but it doesn't code for the entire rest of the virus. So it's really just a piece. It's like if you were just copying or taking the key that the virus has, but nothing else. And that mRNA is then being brought into a regular cell. There's a special type. That actually is a tricky process. It requires some special lipids, like coating of lipids, which are fats, that allow this artificial mRNA to enter a regular cell. And then it's in the cytoplasm, like in the step 2 area of what I just described for the protein biosynthesis. And now the cell, not the nucleus, which is here, now the cell here, the ribosomes, those tools that is essentially eventually create a protein, they consider this mRNA to be just normal mRNA. They don't know that this was brought from outside. So what they do, they actually reproduce the spike protein. And by doing so, the spike protein gets distributed in the body. And then our regular immune response comes into play. So our body recognizes, like in any other vaccination or in any other case where there's a virus, independent of vaccinations, the immune response or the body realizes, okay, we have to defend ourselves because there seems to be a viral attack because we saw this weird spike protein showing up. So then there's antibodies being built and there's T cells attacking these cells here, meaning that for a real infection, at some later point potentially, the body has already all the tools necessary to fight this virus, to fight the real virus. So what the mRNA vaccination does, it essentially fakes a viral attack by only using an artificial and actually also harmless piece of a virus. And it makes the body and the cells believe that there was a real viral attack, but there isn't. And that may also, sometimes potentially be responsible for some side effects, in the beginning of the vaccination, because the body actually just is arming up and thinking there's an infection, and that way some people might have side effects in the beginning, but essentially there's nothing dangerous here. So how does that actually differ from traditional vaccinations? The differences to conventional vaccines, simply speaking, there's a lot more details, of course, is that we can, as when we use mRNA vaccinations, we can generate our viral sequence on the computer, and then just create our mRNAs in the lab. This is way easier than growing real viruses, for example, in chicken eggs on mammalian cells, which is what you have to normally do in order to have enough material for generating vaccines. And also, of course, in contrast to the traditional methods, no real viruses or pieces thereof are being introduced into the human body. So this is actually a fairly safe procedure. It is something where you can really control what is happening. And also to say something about a widespread conspiracy theory that has nothing to do with conventional vaccines, but also with nothing to do with the mRNA vaccination. Well, there is no alteration of human DNA when you use mRNA for vaccinating, simply because of those two-step procedure that I showed earlier. Let me just go back to that. You have this here, and we're only dealing with that part. So this part here is what the mRNA vaccine does. So it introduces an artificial mRNA that is then being translated to create these spike proteins to make the body believe there's a viral attack, and so that antibodies are being generated. But nothing is happening here. Actually, that's also impossible. I mean, even in the regular cycle of our cell metabolism, it only goes this direction. It doesn't go that direction. So there is no way that an mRNA vaccine can alter our real human DNA, which is, of course, a very valuable thing that we don't want to be harmed. And from that perspective, overall speaking, mRNA vaccinations are a great new invention. They're also actually cheaper than traditional vaccines. And they also can be produced just much faster because you don't need like chickens or other things, which take you a very long time often to generate all the amount that you need. And that is especially a problem when you have like a dangerous pandemic like right now. So, okay, I hope I was able to explain this a bit, and bye-bye.